Good day, my friends. I welcome you back to our channel. Today, we will continue for the part 2 of our topic will be cultivation of viruses. So the question now, what or which are the ways scientists they take cultivate, reproduce, or produce viruses for inside the laboratory? Now, we don't talk about animal inoculation before for our video. If you check out that video. Now, this second method we will discuss today. Now, the use of what they call embryonated chick egg. This one mean normal egg wey chicken dey lay. We dey use that normal egg take cultivate virus for many different ways. Now, the first scientist wey been try this method na a scientist wey we dey call Good Pasture, and this scientist been use this method as far back as 1931 to use to produce or replicate many viruses. This one dey important to know. Now, this method dey use chick egg wey be between 7 to 12 days old. Now, we must always remember say in order to cultivate viruses, usually scientists been need any place where the cells they grow and they divide very rapidly. So chick egg or embryonated chick egg na example of such a place, and na im make scientists dey use this chick egg as a means to use cultivate viruses. Usually the chick egg wey dey use na chick egg wey be about 7 to 12 days old. Once that chick egg don dey for about 7 to 12 days old, we fit use that chick egg to cultivate different types of viruses. And wetin be the process? The process simply involve scientists go use iodine. They will use iodine take clean the whole surface of the shell. So that way they wan decontaminate the surface of the cell, make sure say nothing dey grow for that surface of the cell. They go come use a small metal borer. They go use am create hole for inside that egg. Once they don create hole for inside the egg, they go inoculate or put in this virus for inside that egg. Once they don put in the virus for the egg, they will carry the egg like that, put for wetin they call incubator. Incubator na machine wey dey sustain or keep temperature at a particular level. So we go come set that incubator, set am to 36 degrees Celsius. Now, as e don dey 36 degrees Celsius with the egg wey dey inside, and that egg we don put in the virus wey we wan cultivate for inside the egg, we go come leave am for 2 to 3 days. This na the incubation period, the incubation time. After 2 to 3 days, we'll bring out the egg. We'll come notice say the viruses go don grow for inside all the different cells of the embryonated chick egg. Now, after these viruses don replicate for inside this place, we'll come go through the method of purifying the viruses. This method na to separate egg cells from the viruses so that we go fit study all these different viruses. This one dey very important to know. Now, e dey important to also know say for inside the chick egg get very many parts. We don't know say if you look at normal chick egg, yolk dey, many different parts dey, and all these different parts you fit inoculate viruses into all these different parts, and the viruses they go dey replicate, they reproduce for inside all these different parts. Some of the parts wey dey for inside this embryonated chick egg include our allantoic cavity, yolk sac, amniotic sac, and choroallantoic membrane. All these places na different sections within the egg where we fit dey grow our viruses, replicate our viruses for all these different parts of the egg cell. Now, weight of the egg, chick egg. So now, wetin be the advantage of using this chick egg? E get some many advantages. Some of the advantages na say e dey cheaper to use this method. This method dey less laborious, e no dey difficult to do. This method dey na sterile method, so anything like bacteria no go contaminate this whole process. These na some of the examples. E dey cost effective, e no dey too expensive, and eggs dey readily available. So this method na a popular method wey scientists dey use to cultivate viruses. Now, wetin be major disadvantage of using this method to cultivate viruses? The major disadvantage of this method na say different viruses dey grow for different areas of the egg in different ways. Meaning say sometimes if you carry a particular virus, put am for one side, e fit no grow for that side, e no go do very well, e no go reproduce very well. So sometimes e dey difficult for scientists to know the exact area where they go inoculate the virus put for inside the chick egg. So this one na disadvantage, na very big disadvantage, because if you no know the particular area, you fit inoculate for one area and make the virus no replicate for that area. So this one brings us to the end of our discourse on the use of embryonated chick egg to reproduce or replicate viruses. This one dey important to know for science. Thank you for listening. Make una like our content, share our content, give it una friends, and have a wonderful day.